Good morning, everyone. I want to extend a warm welcome to you all and thank you for joining us today for the Restoring Nutritional Landscapes workshop. We hope everyone has viewed the pre-recorded keynotes prior to today as they set the stage and provide important background information for the topics, themes, and projects we'll be discussing. If you haven't yet, you can watch them after the workshop by visiting the keynote page on the Restoring Nutritional Landscapes website. Before anything else, I want to start with an acknowledgement of country. Restoring Nutritional Landscapes is proud to acknowledge the traditional custodians of the places where we work and live. We recognize and respect the enduring relationship they have with their lands and water, and we pay our respects to elders past, present, and emerging. I am humbled to be in the presence of people whose cultures are among the oldest continuing cultures in human history. The first session today is applying what we know about the nutritional ecology of marsupial folivores to landscape management. To provide some background on myself, I'm a landscape and nutritional ecologist in the Finner School of Environment and Society at the Australian National University. I hold a PhD in ecology from the ANU and a Master of Science in Wildlife Ecology and Conservation from the University of Florida. My main interests are in applied ecological research to inform and improve wildlife conservation and landscape management decisions. My colleague, Dr. Karen Marsh, leads the Nutritional Ecology Lab in the Research School of Biology at the Australian National University. Karen completed a PhD on the nutritional ecology of eucalypt feeding marsupials at the ANU in 2006. Since then, she has worked on a variety of research projects, including why captive possums and koalas are picky eaters, how variation in the nutritional quality of eucalypts in the landscape affects the distribution of eucalypt-eating marsupials, and how hot weather affects food quality and diet selection. She is currently investigating how fire and other disturbance events alter habitat quality and landscape use by koalas. In this first session, Dr. Karen Marsh and I will discuss applying what is currently known about the nutritional ecology of eucalypt leaf-eating animals to revegetation practice. We will also talk about important directions for future research to fill critical knowledge gaps in this area. Following this, we will open up the session to Q&A where we will answer your questions as well as discuss real-world challenges and opportunities for restoring nutritional landscapes. We're both very excited to be here. Thank you again for joining us. We'll start now with our presentation and I will hand it over to Karen.
Hi everyone, I just got to get my screen sharing. All right, hopefully everyone can see that. So good morning, my name's Dr. Karen Marsh, and as Cara mentioned, in this first session today, we're going to be talking about ecological applications of nutritional ecology. So we thought we'd start today by briefly going over some of the concepts of eucalypt leaves as food that we introduced in the online keynote presentations. And that way, anyone who hasn't had a chance to watch them should at least be able to pick up on some of the background and the terminology that we'll be using today. Cara will then talk more about using nutritional knowledge to restore forests for folivores. And we'll finish up with a short section on what is missing and where future research needs to be directed. So for this first section, I'm going to briefly talk about nutrients and secondary metabolites, variation in these constituents within and between eucalypt species, herbivore deterrents that are specific to eucalypt subgenera, and how and why the nutritional quality of landscapes can vary. So all of those things are covered in more detail in my keynote presentation on the workshop website. So feel free to go back and watch it again if you find anything confusing in this morning's talk. Now, despite the fact that there are many, many different herbivores out there with voracious appetites for plants, landscapes are usually green. And that's because plants have a variety of physical and chemical defenses that help to defend them against herbivores. So chemical defenses are often called plant secondary metabolites, and they can help to defend plants either by tasting bad, such as being spicy or bitter, or by reducing the digestibility of nutrients so that a plant is less valuable to a herbivore, or by being toxic. So animals need to obtain a variety of different nutrients from plants to meet their nutritional requirements, but plant secondary metabolites can sometimes either deter them from eating a plant or stop them from digesting the plant nutrients. So that means that the nutritional quality of a plant as food is influenced by both the nutrients and the secondary metabolites in that plant. Protein is a really important nutrient for a whole variety of reasons, including playing an important role in growth and reproduction. However, eucalypt leaves contain quite low protein concentrations. And on top of that, some eucalypts contain high concentrations of tannins, which can reduce protein digestibility for some folivores. So the amount of protein that is available to an animal is not necessarily the same as the total amount that is present in the plant. So when you hear the term available protein or available nitrogen, because nitrogen is a proxy for protein, it's talking about an integrated measure that takes into account the total amount of protein in the leaves, as well as the capacity of tannins in those leaves to bind to protein. The fact that the concentrations of available nutrients and plant secondary metabolites work together to influence food quality for folivores means that nutritional quality is not fixed. It can vary both between eucalypt species and also between individual trees within species, depending on what and how much is in a particular tree. Now, we often tend to lump all trees of a species together and think of them as being the same, such as either being an important browse species or one that's not browsed at all, but that's not necessarily how the animals see things. But before I go more into that, I need to remind everyone about two groups of secondary metabolites in eucalypt leaves that are particularly important to eucalypt folivores. Eucalypts in the monocalypt subgenus of eucalypts, which includes species like the peppermints, the ashes, and the stringy barks, produce a group of secondary metabolites known as the UBFs, while eucalypts in the Cynthia myrtle subgenus, which includes a lot of the gums, the boxes, and the ironbarks, produce compounds called FPCs. So different eucalypt species can have different types of herbivore deterrent secondary metabolites depending on which subgenus they're from. And these different compounds can deter some herbivores more strongly than others. So that means that some folivore species tend to eat more eucalypts from one subgenus than the other. So ringtail possums, for example, eat a lot of monocalypt species, while koalas eat more Cynthia myrtles. As well as variation in the types of secondary metabolites between the subgenera, you can also get variation in the concentrations of these compounds. So each of the boxes on these figures here is a different species with UBF concentrations measured on the left and FPCs on the right. And if you look at the two species outlined in red here, you can see that the one on the left has a much higher UBF concentration on average compared to the one outlined on the right. But also even within those species, you get quite a range in concentrations across the individual trees that were measured as part of this particular study. And you can see that you get the same sort of variation between and within species if you look at the species in the FPC figure as well. And this variation has really important consequences for marsupial folivores. Now, this figure here is a feeding study with captive koalas. It shows how much koalas ate when they were offered leaves from different individual manna gums, which is a species that we tend to think of as being an important browse species for koalas where it occurs. Each of the points on this figure is a different tree with a different concentration of FPCs. And you can see that koalas ate much less when trees had high FPC concentrations compared to when the concentrations were low. So variation in the chemical composition of trees creates variation in palatability for eucalypt folivores. with palatability referring to the amount of leaves that folivores eat from a particular tree. Now, eucalypt woodlands and forests are not a buffet of equally palatable food for folivores. They're patchy landscapes. And to illustrate that, the circles in this image are manna gum trees in a 7-hectare woodland on Phillip Island, and each different colour is a different level of palatability to koalas. So within their home range, the suite of trees that are available to koalas can range from highly palatable through to fairly unpalatable, even within a single species. And in this second image on the right, that one's at a much larger scale, and it shows you how you can get some areas within a contiguous eucalypt forest landscape where the available protein concentration is much, much higher than in other areas. One of the implications of living in a landscape with patchy food quality is that you can get variation in breeding success between individuals depending on where they live. So, for example, female brush-tailed possums living in higher quality home ranges have higher breeding success and their pouch young grow faster. We'd expect something similar to happen with the other eucalypt folivores as well. Another implication is for the density of animals that the landscape can support. So this figure shows how the density of koalas at 75 sites across their range is related to differences in food quality. You get higher densities of koalas in areas with higher concentrations of available nitrogen and lower concentrations of FPCs. Oops. So this variation in chemical composition that we see in the landscape is caused by a variety of factors, some of which we don't fully understand yet. However, there is a strong genetic component with secondary metabolite concentrations being highly heritable. There's also an environmental component with factors like elevation influencing some nutrient and secondary metabolite concentrations. Also, if the type of leaves on a tree change, such as when epicormic leaves regrow after fire, nutritional quality can also shift, and changing the tree composition in the landscape through disturbance can also alter the nutritional quality of the landscape. So in summary for this little overview, eucalypt leaves contain a variety of nutrients and plant secondary metabolites that influence food quality for eucalypt folivores. The specific combination of chemical constituents means that some individual trees or eucalypt species are more palatable than others. Leaf nutritional composition is influenced by both genetic and environmental effects. And this means that landscapes are chemically complex mixtures of a range of palatable through to unpalatable trees, which can affect the distribution and abundance of folivore populations. All right, I'm going to hand over quickly again to Cara.
All right, now that we've gone over a quick review of some key points about eucalypt leaves as food, we're ready to start talking about how we can apply that knowledge to restoration and revegetation practice. There are a number of points that I'll cover in this section. This will be followed by a discussion on what we still don't know and what research is needed to figure those things out. Today we are talking about forests as food for folivores. However, folivores are just part of the picture. There are multiple functions of forests, and restoration should consider the influence of composition and structure on more than just food and shelter for leaf-eating animals. However, our experience and our expertise are in eucalypt folivore systems, and so that is the focus of our advice here today. We are very keen to work with restoration ecologists and experts in other areas of forest and wildlife ecology more in the future. This is important for developing a more holistic understanding of how food for folivores fits into the larger picture of healthy, resilient forest ecosystems. Natural landscapes are usually heterogeneous. They have variation even if you can't always see that with the naked eye. When planting, it's important that you include a locally appropriate diversity of tree species. Tree species composition is something that you usually can see but figuring out what should be where can actually be quite challenging. This is because many of the forests in Australia have changed in composition due to anthropogenic landscape change, like selective logging, clear felling, grazing, land clearing, prescribed burning, and increasingly severe and frequent wildfires from climate change. Sometimes a bit of research is necessary to work out what should be growing where, and even then it's an imperfect science, and we can only do our best to recreate the composition that should probably be at any particular place in the landscape. Shifting the composition of forests can have profound influences on the nutritional quality of the landscape. Besides species-level diversity, there are other forms of diversity that are also important, but can't always be observed with the naked eye. Even in naturally occurring monocultures of tree species, you would expect to have chemical diversity that can influence the palatability of trees to animals in the landscape. This diversity is important for both plants and animals to support the longer-term sustainability of the environment and the animal populations. That chemical diversity exists within and between species. Since chemical diversity is not a simple thing to assess, later in this section, I'll talk about strategies to try to incorporate chemical diversity into plantings when you aren't able to conduct chemical analyses on seedlings or parent trees from which you collect seed. Pryor's rule states that in mixed species forests, species from the two main subgenres of eucalypts, which are commonly called monoclits and symphyl-myrtles, tend to co-dominate. Notably, some eucalypt folivores seem to have evolved to tolerate subgenre-specific plant secondary metabolites from one group of eucalypts more than another. This specialization in the ability to contend with subgenre-specific compounds has been hypothesized to reduce competition among sympatric species and support their coexistence. In landscapes where monoclits and symphyl-myrtles naturally co-occur, revegetation should aim to maintain the co-dominance of the main subgenre to support folivore diversity as well. Following Pryor's rule in revegetation may also help folivores to find suitable browse after fire, because preliminary research suggests that their diet choices might change when epicormic growth is available, and I will discuss that more later. When I think about trying to improve the nutritional quality of landscapes, I'm always worried about the potential of planting too much of a good thing and ending up with landscapes like this. You definitely can have too much of a good thing, and this can lead to overbrowsing and subsequent starvation of animals and death of forests. This exact situation is playing out in the Otways and Victoria. These forests are full of a tree species that is often considered one of the koala's favorite browse, manigum, also known as Eucalyptus liminalis. The available nitrogen from the trees that we sampled in this area were some of the highest values that we had sampled from anywhere, despite the fact that those trees, as you can see, are growing on really sandy soil. In this situation, there's clearly too much of a good thing in the leaves, and it is not sustainable for the trees or the koalas. However, it's also important to understand that just because you are planting a species like Liminalis that is often considered a preferred browse for target species like koalas and even greater gliders, it might not be good food. That's because there is a lot of variability in herbivore-deterrent secondary metabolites, even within the same species. This is critical for revegetation because you need to be aware of not just the species you are planting and that those species are appropriately diverse for the environment, but also that you have genetic and chemical diversity. The genetics of a plant can be a strong driver of palatability. The variation that you see in the herbivore deterrent concentrations of the trees in this graph has a strong genetic component. Most of the trees in this graph came from the same area, growing in similar conditions, and yet they show very different FPC concentrations and therefore palatability. It might not be feasible to conduct fuller chemical assays on trees or landscapes in which you want to collect seed or browse from marsupial folivores. So how can you get an idea of what is palatable and what isn't? From sampling trees and recording how much the leaves look munched when we get the branches down, we have found that there is a relationship between palatability and the visual condition of tree crowns. That's because many insects are also influenced by the same herbivore deterrent toxins. So if you want to have a better chance at collecting seed from a tree that's palatable, then choose the scrappy looking tree. The same for if you have an animal in care and you need to feed eucalyptus browse. The best looking tree with the biggest bushiest canopy is also likely to be the best defended and the least palatable. That said, you don't really want to be collecting seed from totally leafless, sickly-looking trees, because there could be more going on there than just palatability. And you also don't want your trees overbrowsed if they are super palatable. Like all science, there are a lot of uncertainties and things we don't know. We don't have all the answers about what proportion to plant tree species and trees of various chemical compositions. We don't entirely understand how to best reproduce the type of heterogeneity that arises naturally. There are genuine risks in planting too much of a good thing or too little. Chemical assays are, for the most part, still time-consuming and relatively expensive. So it's often not possible to know exactly what you're planting from a chemical perspective, even if you did know exactly how much, where, and what to plant. However, like a painter recreating a great landscape with a paintbrush, there are steps we can take to try to reproduce what nature does by using it as our template. Look locally. Identify nearby areas to your restoration project with healthy populations of target folivore species. The animals are there because they can eat the food. If at all possible, collect seed from trees in those areas. Since we know there is a strong genetic component to palatability, even though seeds might not grow into trees with the exact same chemistry as their parents, you probably have a much better chance of ending up with similar palatability than you would with seed from totally unrelated trees or from seed collected from areas where you don't have the target folivores that you're looking to conserve. If you are not able to source seed from those areas, then at least try to use seed from a diversity of parent trees that have been collected from the same region and from a similar elevation. Definitely don't source all your seed from a plantation of trees that have been selected for their vigor and beautiful growth form. Those trees could very well be vigorous and grow beautifully because nothing can eat them. That was actually an option presented to me when I was sourcing seed from seed stores for a restoration project, and it immediately rang alarm bells based on what we know about lush-looking eucalypts. Collecting seed isn't always as hard as you might imagine, and you don't always need tree climbers or sharpshooters. Check out our tutorial videos and published methods on how to use an arborist throw line launcher to collect seed and leaves, if you need them, from a variety of eucalypt trees and for branches up to 40 meters high. Determine the proportion of different tree species and habitat areas that are supporting healthy but not overabundant numbers of your target species. Use those proportions as a guideline. Consider scale. Forest composition is rarely consistent across wide areas. When determining tree species composition, sample multiple points across the site that you are seeking to replicate, and sample across a similar scale to the area that you are planning to restore. Be aware of how changes in topography, aspect, and riparian areas correlate to natural changes in tree species composition, and let nature be your guide. What about water? Many eucalypt folivores get most of their water from the eucalypt leaves they eat. Are eucalypt trees better food for folivores if they're closer to surface water? This is a good question. There are some eucalypt species that need a lot of water and they don't do well when they lose access to it, like river redgum. However, we don't have any evidence in most places that surface water is an important feature for food quality for most eucalypt folivores. In addition, we have not found any effect of topography on leaf moisture. For example, comparing trees growing on slopes, ridges, or flats and full of moisture content in the places where we have investigated this. That doesn't mean it doesn't happen anywhere, but we haven't seen it. Most of the eucalypts we have sampled have between 40 and 60% moisture content. If animals can eat enough leaf, they should be able to meet their water balance. Most eucalypts seem to be very good at maintaining their fuller water content because they have very thick leaf walls that help them avoid desiccation. In a recent study, we found only 3% difference in eucalypt leaf moisture from trees sampled on the north coast of New South Wales between drought and non-drought years. That's not to say that severe drought cannot result in a loss of leaf moisture below which animals and the plants themselves require, but this would be an extreme situation and probably more likely to happen the further west you go. Drought often occurs with extreme heat events, and this heat, as we learned from Dr. Pitfields' keynote, results in animals eating less. When animals that depend on foliage for their water eat less, they are also more likely to get dehydrated. Therefore, rather than eucalypts not having enough water, which is probably not as common as people think. Animals may simply not be eating enough to get the water they need because they're getting too hot. In these circumstances, access to free water might help. However, some species like greater gliders have never been seen to come down from the trees to water bodies at all, so it's unlikely to make any difference for them from that perspective. However, I'm saying all of this from a perspective relating specifically to food quality. Surface water like creeks and rivers can substantially alter microclimates, making areas with water more likely to remain cooler through the evaporation of the water source. This could help animals persist in these areas, particularly during heat waves. Can we drought-proof these landscapes? In extreme drought, even tough eucalypts will desiccate and die. Tree species that require more water, like river red gum, really struggle during drought conditions as well. When the trees die, the folivores in those landscapes will die too, and probably a lot of other things. Is there anything we can do from a revegetation perspective to help these landscapes persist in a drying and warming climate? This is another very good question, and people are already looking into genes that make eucalypts more drought tolerant. There are trials to grow more drought-resistant forests and use climate-ready plants for revegetation, some of which are already available. This all sounds really promising. However, and this is a very big however, some of the secondary compounds that make eucalypts less palatable to folivores may also confer protection against climate extremes. We do have some evidence that PSMs, like FPCs, help leaves withstand higher levels of solar radiation. It is common for secondary compounds to serve multiple purposes. We need to be very careful that we are not selecting for a specific climate-ready property at the expense of something else like palatability. Otherwise, we risk having very green forests that can resist drought, but also resist possums, gliders, and koalas just as well. It's critical that the genotypes in trees being selected due to their resistance to heat and drought are also analyzed for their concentrations of plant secondary metabolites that can deter herbivores. This has not happened yet, and it really needs to before these trees are rolled out en masse. We are looking at the system through a very narrow lens if this is the only thing we are considering. If the only thing we are considering is the tree's resistance to heat and drought. From a folivore's perspective, a tree might as well be dead if it's not edible. Recent research into the effects of fire on browse quality has also found that the composition of epicormic growth in the subgenre differ in their nutrient and herbivore deterrent compounds from mature leaf. These differences can be substantial and result in different browse choices by eucalypt folivores when epicormic growth is present and mature foliage is scarce. This means that eucalypt folivores may even switch the type of trees they eat after disturbance based on the type of foliage available. Trees that they could eat the mature foliage from, they might not be able to eat the epicormic growth, and vice versa. In addition, very early results from research on the north coast of New South Wales and the southern tablelands suggest that fire has very different effects on the nutritional quality of monoclips compared to symphymertals. For animals that are more reliant on monoclips like ringtails and to some degree greater gliders, fire may have a stronger negative impact on nutrition than for animals that usually eat more symphymertals like the koala. Although we have only started to scratch the surface with understanding the impact of fire on nutritional landscapes, it is clear that where there is naturally occurring diversity of eucalypt species, that should be replicated in revegetation. In addition, adhering to Pryor's rule when it's appropriate, that is that monoclips and symphymertals tend to co-dominate when multiple tree species are present, may help some eucalypt folivores find food after fire. So this might seem like it goes without saying, but a surprising number of revegetation initiatives, particularly for carbon offsets, are planting trees in landscapes where they don't belong, like savannas and prairies. This comes at a cost to the local native ecosystem and biodiversity more broadly. Many of these areas also experience frequent fires and less moisture than most forests require, so it's a bad long-term investment strategy anyway. Next point is that just because trees will grow somewhere doesn't mean that an area can support the folivores you're trying to conserve. It's important to consider whether that area is in the climate envelope of the target species. If it gets too hot or too cold, that could make it uninhabitable in the long term, even if the right trees are there. What other threats are present in the landscape? Feral dogs, a lot of vehicle traffic, lack of connectivity to other areas of habitat, these things can all preclude the use of your restored forest by target species. Also, as mentioned previously, although many species distribution models predict that animals and plants are likely to move upward in elevation due to climate change, trees growing at higher elevation can be deficient in the central nutrients. More work in this area needs to be done before we can safely assume that these animals will be able to have suitable homes higher up in the mountains. Don't assume poor soil equals poor forest for eucalyptolivores. Don't rule out locations for forest restoration just because the soil is what we would normally consider infertile. Some of the best nutritional quality trees that we have measured for eucalyptolivores appear to be growing in areas with low fertility soils. Healthy populations of eucalyptolivores are not restricted to fertile alluvial plains. After all, you're not farming wheat in Kansas, you're growing eucalypt forests in Australia, and these trees to some degree are very well adapted to the environment in which they live, and so are the animals. And don't plant delicious trees where you don't want animals. This includes roadsides, verges between busy roads, and timber plantations, and anywhere else where animals are likely to be injured or killed if they try to access that patch of delicious plants. Plant poor quality food in those areas, and that could be a very useful strategy actually for reducing harm to wildlife and potentially reduce things like vehicle collision. Now I will turn this back over to Karen so she can talk to you about directions for future research.
Okay, hi again everyone. So last section of this morning's presentation is about what is missing from the overall picture. So I'll talk a bit about some of the essential research that needs to be done to fill critical knowledge gaps, and then Cara will talk about improving accessibility to nutritional data with some of the up and coming technology in this space. So one thing that we definitely need to look into is how the chemical heterogeneity of restored habitat compares to natural intact forest. So we know that secondary metabolite concentrations are heritable, so the way that people source seeds for revegetation projects could potentially influence the chemical diversity of the restored landscape. So, for example, if the seeds for each species are sourced from only a small number of trees that happen to be fairly unpalatable, you could end up growing a landscape that is never going to have the capacity to support eucalyptolivores. Or, on the other hand, you could have a situation where every tree is so nutritious that you end up with issues with overbrowsing and unsustainable folivore populations down the track. So there's a need to undertake research to see how these restored habitats compare to natural forest and think about how we can adapt our practices to more closely mirror what's happening in local natural systems. Another interesting area for potential investigation is whether targeted planting of high quality trees is an effective strategy to improve the overall nutritional quality of forests and potentially increase carrying capacity of the landscape for folivores. And that's an interesting idea and it's got a lot of merit, but it really needs some long-term research investment to see if the outcomes of this strategy are what we would expect and or like them to be. And a very important area where more research is needed is how some of the environmental factors influence nutritional quality. And that's because we need to know how much the planting location is going to influence tree nutritional composition and suitability for folivores. So the biggest questions here are to do with soil, climate, and elevation. For the soil question, there's this widespread assumption that soil and leaf quality are linked, mostly because the two are related in agricultural crops. But we think that that might not always be the case in eucalypts. And Cara already mentioned locations where we have sampled eucalypts in what would normally be considered a low fertility area, and they've been quite nutritious for folivores. For climate, we know that animals eat less when it's hot and that they're more susceptible to secondary metabolite toxicity in heat. And Pip Beale talked about that in her keynote presentation. So we need to know more about how important it is for folivores to have access to eucalypts with lower secondary metabolite concentrations in hotter weather. Also, what role does nutrition play in determining climate refugia for these animals? And of course, we need a better understanding of how elevation affects leaf quality, since many animals are predicted to shift their range upwards in response to climate change. And certainly our preliminary studies suggest that food quality may be poorer at higher elevations. If we want to regenerate habitat that has the best chance of supporting folivores in the face of future stochastic events like fire and drought, we also need to know more about their effects on food quality. So short-term changes in leaf types, such as post-fire epicormic regrowth, can alter available nutrient and secondary metabolite concentrations, so that species that were highly palatable will suddenly become less palatable and vice versa. We really need more research in this area to understand how important it is to have a diversity of eucalypts available that produce high quality food under different scenarios. Another factor that we have limited understanding of at the moment is whether changes in the availability of water and/or nutrients for plants in response to drought or fire affect the production of some of the chemical constituents. And disturbance can also change species composition. We need a better understanding of how the change in eucalypt species composition in response to drought and fire affect the nutritional landscape. And again, as Cara mentioned earlier, we also need to understand whether there are any unintended consequences of selecting for drought-resistant trees when we want restored landscapes to be able to support folivores. We know that secondary metabolites often have multiple purposes in plants. They're not just there to protect the plants only from herbivores. So it could be that plants that are more resistant to drought are also more resistant to herbivory. And the last piece of missing information that I'm going to mention is whether there is a link between nutrition and the health of folivore populations. So it's really well known in domestic animals and in humans that individuals in a better nutritional state are more likely to be healthy and to be able to fight off disease. There's a number of diseases in eucalypt folivores, some that are particularly well known in koalas, where there are known differences in the prevalence and or the severity of disease between populations. We don't currently know whether any of these differences are related to the nutritional state of the animals and the forests in which they live. From a management perspective, it would be really valuable to understand whether you could carefully plan revegetation projects in a way that would help to support the health of eucalypt folivore populations. Okay, so handing over to Cara for the last bit.
All right, hello again. Our last topic is how we can improve accessibility to nutritional quality data and the type of research that needs to happen so more people can use this information if they want to. Information on the chemistry and nutritional quality of plants can help us assess habitat quality for many different species and is particularly useful for understanding the ecology, habitat requirements, and value of particular areas for iconic Australian species like koalas and greater gliders. There are currently only a couple of labs in Australia that are able to analyze eucalypt leaf samples for digestible protein, also called available nitrogen, FPCs, and UVFs. We already cannot keep up with the request for this type of information from a variety of different user groups. Interest and demand is only getting larger as the value and application of this type of data are becoming more widely apparent. So how can we make it possible for more people to have access to nutritional quality data? And how can we assess nutritional quality across wider areas? I used to think that airborne hyperspectral imagery was the answer. It's very cool and it can estimate the nutritional quality of eucalypt foliage from spectrometers mounted to aircraft. I've done it for my own PhD. That's where this image of available nitrogen in eucalypt forest landscape came from. Others with whom I've worked have also estimated FPCs using the same technology. However, when I tried to do this again across wider areas in New South Wales and Queensland, I couldn't. Not because it wasn't possible, but because at the time, about 6 years ago, a number of factors aligned that made it unachievable. Part of it was me biting off more than I could chew and trying to do both large-scale land-based nutritional quality assessments and airborne nutritional quality assessments at the same time. And part of it was having amazing remote sensing facilities and capabilities defunded at CSIRO and losing access to a lot of the support and infrastructure and people that make this kind of work possible. And those are the real consequences when our governments defund science. Making nutritional quality maps from airborne spectrometers seems like it should be a matter of just flying some planes or drones over a forest and then pressing a button and a map pops out. In reality, though, there's a lot of processing steps, ground truthing, actual lab chemistry, developing training and testing data, pre-treating, denoising, transforming spectral data, applying these transformations to huge multidimensional data sets, which require supercomputers, and all of this takes a lot of time and highly skilled, specially trained people across multiple fields to achieve. There's no software program that exists that we can enter this data and simply get an easy, useful output by pushing a few buttons. I still believe that airborne and satellite-based spectroscopy will eventually be able to deliver what it promises, and is already doing cool things. However, we need dedicated teams of people to write programs and create graphic user interfaces for automated large-scale spectral data selection, transformations, and processing that work in concert with supercomputers, are able to substantially reduce the dimensionality of the data so that we can process the information and create the outputs on our own laptops by clicking a few buttons. Then we can all enjoy and use the amazing chemical maps that they produce. Drones are somewhat similar in the way that they collect data to airplanes and satellites, except that they usually fly much closer to the trees and don't cover as much area. This can help make processing somewhat simpler, but the spectrometers that drones carry often not have the shortwave infrared wavelengths that cannot some of the herbivore chemicals to look at. Sasha, are you able to mute Kat?
Yes, I'm trying to, sorry.
Thanks. So anyway, that's changing rapidly. These sensors are getting better that drones can carry, and they hold considerable promise for eucalypt nutritional quality mapping in the future, but it's still not a simple matter and is going to take a lot of research to achieve. It's also worth thinking much smaller to achieve big things. Handheld spectrometers have the potential to allow us to take what is traditionally laboratory equipment to the field to make measurements of plants in situ. These are a step up from the slightly larger field versions that are already routinely used to assess things like pasture quality for grazing herbivores. After all, what we're doing is very similar. It's just that our pasture is very tall and our cows live in the trees. I was amused to discover that even commercial pot growers can use these portable spectrometers to determine the THC and cannabidiol content of their crop, which after all are just plant secondary metabolites too. I figure if pot farmers can do this, then surely this technology can be used by anyone. No offense intended to the pot growers. That said, there's still quite a bit of work required to set up the calibrations that are needed for these spectrometers to do what we want them to do. I'm optimistic that it will be easier than trying to map fuller chemistry from airplanes, but it will still take some time and funding to properly develop this technology for our specific application. I really think it has a lot of potential though, and we can learn a lot from our friends in the agricultural industries. In fact, even the techniques that we use now in our laboratories to assess nutritional quality of eucalypt leaves have their foundation in agricultural science. So that's the end of our presentation today.